Hi, welcome back. We're going to talk about Chapter 5 Vector Control. Okay, so uh, what is the difference between a causative agent and a vector? Okay, causative agent is the one that causes a disease. It's a protozoa, viruses, bacteria, or um, prions, or rickettsia, and so forth. Okay, so these are causative agents which causes the disease. Okay, this is clear. Now, what is a vector? A vector is what do you call in layman's language, a carrier or a middle person. Okay, which carries the causative agent into the host, a human host, and then ultimately causing the disease among humans and animals as well. Okay, so that is a vector. Okay, I'll give you some very simple example. Tuberculosis. Okay, tuberculosis is caused by tuberculosis bacteria, as you know. Okay, so if someone has a tuberculosis and you stand next to that person, and if that person sneezes, coughs, then there is a chance that you might get tuberculosis. Why? Because it's tuberculosis is included in a special class of diseases, which is referred as infectious diseases. Okay, so infectious diseases where they can be transferred from person to person from simple things like sneezing, coughing. Okay, but for vector-borne control, vector-borne diseases, it's a different thing. Okay, if somebody has a Lyme disease or malaria or uh, dengue fever, okay, if you happen to be close with that person sneezes or coughs, or if you're in close surrounding of that person, you're not going to get dengue fever or Lyme disease. Why? Because the disease doesn't transfer person to person. It requires a vector to transfer the disease. So if that person has a Lyme disease and a tick that causes Lyme disease is somewhere around there, then that tick needs to bite that person, get those causative agent from that person's blood, and then insert that causative agent into you, and then you can get Lyme disease. So what I'm saying is this: when you this involvement of vector is a key area in vector-borne diseases. That's why they're called vector-borne diseases. Diseases that are carried by vector. In absence of vector, even though a causative agent is present and host is around, the disease transmission will not happen. This is a very, very important thing about vector-borne diseases. Now, there are a very, very important significance of vector-borne diseases. There are two types of vectors. Okay, one is insect vectors. Insects are mosquitoes, ticks, biting midgets, and flies and things like that. And other type of vector are larger animals, vertebrate animals, cow, dog, cat, horses, rodents, all these animals. Okay, uh, so these special class of vectors are referred as zoonosis, so diseases that are caused by animals. Okay, they're also vectors, but they're animal vectors, and the other ones are insect vectors. Okay, so uh, that's the difference. Now, in terms of insect vectors, usually a biting is required. Biting meaning they bite you and insert their trunk into your skin. It pierces your skin, and the trunk pierces the capillary and sucks the blood, and at the same time it inserts the causative agent into the blood directly in the blood circulation. Okay, so biting is required for insect vector. For animal vector, biting may not be required. Okay, so when if the person is in a immediate surrounding of an animal, where the animal's waste products are there, urine or dung, or its food products are there. So in that immediate surroundings, this causative agent could be present. And if the person is nearby, those nearby that environment, even from uh, e- even inhalation, even if the person if person is there, he or she can be exposed to the causative agents and can acquire the disease, okay, through inhalation or other things. Okay, for example, leptospirosis, okay, it transfers through inhalation of droplets of urine of a rat that is infected with the leptospirosis bacteria. Okay, so the key idea for a vector in general is that, except for the most part insect vectors, is that the immediate surrounding is important. Biting is not required. Okay. Of course, this depends case to case. For example, rabies, the rabies infected dog or animal needs to bite and needs to introduce rabies virus into the blood circulation. Otherwise, rabies will not happen. But in anthrax, just inhalation of spores from cow dung, from the cow that was infected with anthrax, person can get anthrax. So it depends on the a type of disease. So the key idea is immediate surroundings. Okay. Uh, so hoping that this idea is clear. Now moving on to the different types of diseases. Okay. Diseases are that we discussed here are malaria, and what is leishmaniasis. Um, then we have Lyme disease, Rocky Mountain spotted fever, um, and then viral diseases that are called viral diseases, arboviruses, West Nile virus, Zika virus, Ebola virus, um, emerging pathogens such as hantavirus, okay, dengue virus, and there are other miscellaneous diseases such as rabies, anthrax, and so on and so forth. So you can read that description from your book. Okay. A key idea to remember is that you need to know which is a causative agent. Is it a bacteria? Is it a virus? Is it a rickettsia? Is it a parasite? You need to know that. Okay. For example, for malaria, the causative agent is a parasite. For leishmaniasis, causative agent is a parasite. For dengue fever, causative agent is a virus. For Rocky Mountain spotted fever, causative agent is a rickettsia. Okay. Uh, s- uh, Mad cow disease, the causative agent is prion. Okay. So these are different types of causative agents that you need to remember. Also need to remember a life cycle of malaria. Okay, so uh, why the involvement of vector is necessary? Somebody might ask. The reason is that the the causative organism needs to complete a part of the life cycle in the insect's body, insect's intestines, and other part of the life cycle in another host, usually animal host or human host. Okay, among sometimes in humans, they have to complete some life cycle in liver, some life cycle in blood cells, some life cycle in another organ. So it's a very complicated life cycle of these parasites or these causative agents. So for that, they have to involve an insect vectors to complete their life cycle. Okay, for example, in terms of malaria, the person gets the parasite is Plasmodium. Okay, there are four types of species. Falciparum is the most dangerous. So that that Plasmodium is in usually in a dirty environments, unsanitary conditions, standstill water, where which is also good for growth of mosquitoes. So this environment that is favorable for both parasite and mosquitoes. Okay, so the parasite is growing. The parasite is there, but they are not. They need. They cannot multiply without unless they are in a host's body. Okay, so the mosquitoes gets those parasites from that environment, maybe like having a drink or something, and they're in water, and mosquito gets that, and then the parasite the parasite multiply in the stomach of mosquito, and then the life cycle starts. So they multiply. There is a reproduction. They multiply, and then the final product, the progenies of those that are multiplied, they're like thousands. Okay, now those they erupt out, and then when mosquito bites a person, the mosquito introduces thousands of these parasites babies into the person's body. Okay, once they are in a person's body, these products, which is called as sporozoites, they travel into the person's liver first. Okay, then again the same situation happens. There is a life cycle that need to complete, and they mature and then burst out, and then you can see all this in the book, and then there is another product, and then they travel into the blood cells, and then they mature, and then they burst out. So at every stage, and in liver, they are from thousands to that maybe like multiple thousands, and then from in the blood, they are from multiple thousands to like in millions. That's how their numbers are increasing, and the final product in the blood stage is referred as a gametocyte. Okay, so the gametocyte stage of a parasite is the most dangerous and most infectious. Okay, so when the mosquito can bite the person and inhale those gametocytes, and can transfer into another person. So then in that another person, it doesn't need to go through all this liver cycle again, and there's a ready form of gametocytes are already available. These gametocytes travel in different organs, liver, different organs such as, of course, liver, and also brain and other organs, and they reduce, once they're in blood, they reduce the content of hemoglobin. They affect the RBCs. The RBCs are destroyed, so the person's hemoglobin is very very down, and person have a very severe type of anemia. Okay, also when it's in liver, the person has severe chills. Okay, so you can see the clinical aspects as well. So when it's in the liver stages, when the parasite is completing the life cycle in liver stages, the person has severe chills. When the person is when the parasite in the blood stages, then person has a severe anemia, and which will show in the lab reports, and also clinical findings will be there. So that is what we call as full-blown malaria. Okay, then we need to treat the malaria. We need a quinine drug, chloroquine, mefloquine, these type of drugs which are very very effective for malaria. Those can be used, and can save the person. Otherwise, malaria can kill the people, and there is a huge burden of malaria related deaths, and direct and indirect cost globally. Okay, in US it's less of the risk because the sanitation is good, and very good public health system, but in other countries it's a problem. They are still struggling. Malaria it's a global burden. Okay, this is about malaria. Leishmaniasis. There is also life cycle of leishmaniasis, and that needs leishmaniasis parasite completes the life cycle. Part of life cycle in the sandfly's body, intestines, and part of life cycle in the person. Okay, and it causes a sore that you can see in the book, sore on the extremities where it has the fly has bitten, and then you can see those in the book. And similarly, you can study the life cycle of leishmaniasis parasite. Then we have plague. Plague is uh, caused by bacteria, yes, Yersinia pestis. The vector that is used is the vector that carries Yersinia pestis bacteria is flea. Okay, and that flea is harbored by rodents, which is mysis, and the flea when the rodent is in contact with people, the flea they jump from rat's body onto the person, and then they cause plague. That's how plague was caused in medieval times and have killed many people in those days. And fortunately, we have antibiotics and we have very good sanitation control methods. So we make sure that there are no rodents and no filthy stuff that is going around, and we can have, that's why we have very good control of plague. So globally, there is uh, almost complete eradication of plague, except a few outbreaks here and there. Then we have Lyme disease. Lyme disease is caused by bacteria again, Borrelia burgdorferi. Okay, the complication of Lyme disease is arthritis. Even though a person can be treated with antibiotics, uh, still it leaves a complication. The bacteria is died, but before that it does something to the skeletal system. That's why a person has a arthritis. And that's not just one joint; there's multiple joints, and sometimes it's temporary, sometimes it's permanent. So it depends. So arthritis is a problem with the Lyme disease, and there are different types of ticks that you need to know. Black leg tick causes Lyme disease, and a dog tick, which is a bigger tick, that causes Rocky Mountain spotted fever. Okay, Rocky Mountain spotted fever is caused by Rickettsia. It's a completely different type of creature. It's in between protozoa and bacteria, so it has both of features and can be easily controlled by antibiotics. So that's a good thing about it. So Rocky Mountain spotted fever starts with fever for 1 to 3 4 days, and then there are peculiar rashes are there, and sometimes can be fatal if it's not treated very well. Okay, so we have completed these parasite and bacterial illnesses. Coming to the viruses, okay. Viruses, we have arboviruses, so arthropod-borne viruses. Uh, so the viruses are very small creatures. They are undead particles. So yeah, they are dormant, which means inactive for a very long time in the surroundings. They become active when they are in host's body. They multiply very fast, and they can change their genetic code. So it's that's why it's hard to treat them, and there is no treatment available for many viral diseases. It's just self-control, what's called recovery treatment is there, supporting treatment. Okay, okay. Now there are different types of viruses that viral diseases that you need to know, and mostly they affect nervous system, so central nervous system and other types of problems. Also, a major worrisome factor is the hemorrhages. Some of the viruses like Ebola causes hemorrhages. Dengue virus also causes hemorrhages. So that's how people die from those viral diseases. So that's why it's extremely important that these are the vectors for these viruses, the mosquitoes in most cases, should be taken care of to prevent these type of illnesses and resulting deaths. Okay, now we have emerging pathogens problem. So because of global warming or the climate changes, the areas, the climate is changing, the weather patterns are changing, so it becomes suitable for some of the insect vectors now. So and that's why the diseases that they cause have been on the rise. It is referred as emerging pathogens. Okay, in US, it's a classic example of hantavirus. So you can see how the weather, cold weather, and then the floods and drought situation, and that cause influx of rodents, mice, and then there was a hantaviruses cases that were discovered. You can read from the book. So there are these type of hantaviruses and emerging viruses that you need to know. There are other diseases such as rabies and anthrax are there. Rabies is a viral disease. It's a zoonotic disease. In developing countries, it's caused by stray dogs. In developed countries, mostly chipmunks or other types of rodents or bats. So the virus is transferred to human from biting. Good thing is that we have what's referred as a post-exposure prophylaxis that is there. So once within 48 hours, if the person gets treatment, injections, a person can. There's a 99% survival rate. Otherwise, if that window is gone within 48 hours, that person doesn't take the injection. If there's a delay, then chances of survival decreases. If there are no injections taken at all, no treatment is taken, no post-exposure prophylaxis is taken whatsoever, the survival is 0%. 100% death will happen from rabies. Uh, Anthrax, 25% of cases can, if left untreated, can cause fatal, can become fatal. All the anthrax cases can be treated by antibiotics. So that's the different types of anthrax: inhalational anthrax, cutaneous anthrax, gastrointestinal anthrax, and the inhalation is the most severe form, but all can be treated with antibiotics. Um, and there is influenza that you need to know. There is avian bird flu. Okay, so sometimes the influenza cases where the carriers are basically these mosquitoes, and then uh, they bite the birds, and then the mosquitoes, the pathogens become active, and then they insert into the person, and there is a bird flu, and many people have died from bird flu. There have been some bird flu outbreaks have been reported. I need to know about those. Then the control of all these vector-borne diseases. Um, the best way is sanitation control. Okay, get rid of this standstill water, improve drainage of waters, make sure that there is no safe haven for vectors to grow. That's the main thing. Infrastructure control is very optimal. Then there is a use of pesticides. Used to be a DDT. DDT was very effective until 1940. In 1940s, it's what was it actually was considered as a miracle uh, that the malaria outbreak in U.S. malaria epidemic was huge in U.S. And in 40s and in 49 it was completely gone, and that was mostly because of the action of DDTs. But then DDT was banned. There are other pesticides, so action of pesticides that can be sprayed and they can kill these vectors. So these, and for Lyme disease, use long sleeves, and when you go working on the backyard or going on the treks in the forest, use a long sleeve so that the insect cannot stick on your skin. And long use gloves and use boots and things like that. So basically, what we call personal protective equipment. Those are the measures that are recommended. Um, okay, so these are the preventive measures that can be used. And hopefully, again, there was a long video, but it's a very big chapter. There is a midterm exam that's coming up. So I will post a study guide, and see the study guide, and you should be good. Okay, thank you for this for watching this video, and I will talk to you about another chapter in coming weeks. See ya.